All right, welcome back, everyone. So we're going to pick up here with our kind of transport processes series of videos and begin looking at nutrient reabsorption. And we're going to look at a few in particular. We're going to look at some protein. We're going to look at sodium. We're going to talk briefly about glucose, and then we will move on from there. Right. So first is this idea of nutrient reabsorption. Right. So nutrient reabsorption is moving some type of molecule, right? Some nutrient, some solute maybe, from the tubular fluid, uh, ultimately back into the blood, right? It's not going to go directly from the tubular fluid to the blood because it's got to go either between or across a cell. But this idea of reabsorption is tubular fluid to blood, right? So when we talk about these processes of, we already did glomerular filtration, now we're doing tubular reabsorption, we'll do tubular secretion. Think about the direction that the solutes or the molecules or substances, think of the direction that they're moving, right? Are they moving from the tubular fluid to the blood? Are they moving from the blood to the tubular fluid? Think about that, right? So this process is dependent on various transport proteins, right? So you've talked about in previous lectures and cell lectures, um, you know, various types of transport proteins like protein carriers, protein channels, right? And how these are largely ion specific, right? So there are specific ones that are for sodium and potassium and calcium and glucose and so on, right? And so that's what we're looking at here. So an example here is with glucose transport, right? And so with glucose transport, if you'll focus your attention here kind of on this luminal side of this cell, right? We've got sodium and we've got glucose, right? Well, if you think back to previous lectures you've done um, and been involved in, where is sodium more highly concentrated, right? Where is sodium? Where do you find it more highly concentrated? Inside the cell or outside the cell? Well, it's highly concentrated outside the cell. So if you've got lots of sodium on the outside, and just a little sodium on the inside, then sodium moving from outside to inside is moving down its concentration gradient, right? That's passive transport, right? That's diffusion, all right? What you can then do is you can take another substance, okay? So in this case, it's glucose. So these little green molecules are glucose. And you can move it against its concentration gradient, or you may hear it termed up the concentration gradient by using another substance, in this case sodium, moving down its concentration gradient, right? So this is something called secondary active transport, right? We're utilizing the movement of one substance moving down its concentration gradient to move another substance against its concentration gradient, right? So we move sodium from high to low inside the cell, and at the same time, glucose is moving at the same time, right? So this is something called a sodium glucose symporter, right? And a symporter is a type of co-transporter moving two things at once. And a symporter is specifically one that moves the two substances in the same direction, right? So we're moving the glucose from the tubular fluid into the into the cell, excuse me. And then once we have the sodium and the glucose inside the cell, that glucose is going to use a glucose transporter and it's going to be moving across the basal surface, this kind of basement surface of the cell, ultimately back into the blood. And again, like we mentioned, or we were talking about in the previous video, under normal circumstances, 100% of that glucose is moving back into the blood, right? There should not be any leftover glucose in the tubular fluid, which is ultimately going to be the urine, right? But here you can see how you could potentially have, if you had far too much glucose here, if you had way too much glucose and you didn't have enough of these proteins, then of course some of that glucose is going to get uh, left in the tubular fluid and ultimately end up in the urine, right? Um, so this is again, this is one process by which you can begin to move um, substances from the luminal fluid ultimately into the blood, right? So this occurs with glucose. It's also occurring for things like amino acids, lactate, um, other molecules, phosphate, sulfate, other ions can move via this kind of secondary transport type of process, right? Um, when sodium is reabsorbed, it's not necessarily always moving alongside alongside another molecule, but it can it can be often, right? Because it's going to be a big driving force for other solutes, other molecules, right? So another one we can look at is protein, right? So protein, again, is moving from this luminal surface into the cell and then across this surface. And so this is moving by something called penocytosis, right? So penocytosis um, is a type of endocytosis in which we are then we're bringing in, almost in kind of like eating, if you will. It's endocytosing, in this case, the protein um, from one side to the other, right? Now, in this case, what you see, or what hopefully what you see, is that you endocytose the protein, right? The protein here from the luminal surface. And again, remember, protein is usually not in the urine, right? If it is, that's called proteinuria. But here we're moving things, we're moving protein from the luminal fluid, the tubular fluid, into the cell. And then what's actually moving into the blood, it's not the protein, but it's amino acids, right? It's amino acids. So these proteins are broken down by various enzymes called peptidases. These are enzymes that break down proteins. And then what's actually moving into the blood are the amino acids and not the proteins themselves. Okay. Um, so it's just a different process, right? Now, certain proteins, really large proteins, think maybe like albumin or something, um, they would likely not even end up in the tubular fluid in the first place, right? So we're talking about small proteins, medium proteins, and maybe small amounts of large proteins. But by and large, most of the larger proteins will not even end up in this tubular fluid in the first place, right? We can also look at sodium reabsorption independent of other molecules, right? So previously we talked about the sodium glucose cotransporter, right? You can also just have sodium reabsorption, right? So again, most of this is occurring in the proximal convoluted tubule, right? So here you see in your tubular fluid, right, you've got a high concentration here, a lower concentration inside the cell. So we're moving from high to low, right? Notice that is a type of diffusion, right? In this case, it's facilitated diffusion, right? Remember, facilitated diffusion simply means it's diffusion with the assistance of a protein, right? And so in this case, it's a sodium channel, right? So it's allowing sodium to move through, right? Notice what type of transport is this, right? We already said it's passive, but is it paracellular or is it transcellular, right? Well, this is, hopefully you're saying this is transcellular, right? We're moving across the actual cell here, right? So we're moving from this side across. We're not moving in between, that's paracellular, we're moving across, right? So now we've got sodium on the inside, but think about it, if we're moving sodium to the inside of the cell here, right? So high to low, ultimately the sodium is going to build up, right? And if it builds up too much, then we're going to kind of erode this concentration gradient. So we've got to do something that keeps this concentration of sodium inside the cell low, right? So the way you do that is with the sodium potassium pump, right? So we're using the sodium potassium pump here to pump 3 sodium out of the cell, ultimately into the blood, for every 2 potassium we're bringing in, right? So sodium is moved up its concentration gradient. Again, lower concentration inside the cell, higher outside, so it's pumped, right? Anytime you see that term pump, think active transport, right? So we're using ATP, we're using energy to move that sodium out, ultimately into the blood, and then our potassium is moving back in, right? So sodium and potassium are always going to move in more or less opposite directions, right? Largely because of that sodium potassium pump, right? 3 sodium out for every 2 potassium in, right? Now, this process can be regulated by various hormones, right? And the hormones are ones you should be familiar with, things like aldosterone and ANP, right? So aldosterone, again, for both of these, think about where they're coming from, right? Think about where they're coming from and what their effect is, right? So aldosterone is coming from our adrenal cortex, right? That zona glomerulosa of our adrenal cortex. Remember, you have the glomerulosa, the fasciculata, and the reticularis. This is a mineralocorticoid coming from the glomerulosa. And what it's doing is it's stimulating these sodium channels and sodium potassium pumps, essentially to increase this process here. So this process we looked at on this slide, aldosterone is acting to further enhance that, right? Put in more sodium channels, put in more sodium potassium pumps. So you can bring in more sodium into the cell. You can pump more sodium out of the cell, ultimately into the blood. Uh, so increasing sodium reabsorption. And then following the sodium, we have water, right? So remember, sodium, it's going to exert an osmotic gradient, right? So as you pump more sodium out into a given region, some given space, some given area, it's going to increase the concentration of the solutes in that area, so of the sodium, and water is going to move in that same direction in an effort to equalize the concentrations, right? So water is going to follow. So general rule of thumb, anywhere where you have sodium going, water is going to follow it, right? And in a little bit, in kind of one of our next parts, we're going to talk about different types of water reabsorption, okay? But we won't get into that right now, right? And if you want to know, this is occurring specifically in cells called principal cells of our collecting tubule, collecting ducts, things like that. Right? Now, the other hormone is something called AMP, atrial natriuretic peptide. We talked about this in, I think, in our last series when we were talking about glomerular filtration. We've talked about it when we did heart and blood vessels. I think we talked about it when we did endocrine system. We've talked about it several times, right? So essentially, think of this as doing the opposite of aldosterone, right? So it's going to inhibit the reabsorption of sodium. It's going to inhibit the release of aldosterone. So all these things that aldosterone is doing, ANP is doing the opposite, right? So aldosterone is acting to bring ultimately more fluid by way of sodium, but more fluid into the blood, right? Well, atrial natriuretic peptide, remember, why is it released? It's released in response to increased stretching of the heart, right, of the atrium. Well, why is the heart stretching? Because there's excess blood volume, there's high blood pressure, right? So in the case of increased blood volume and blood pressure, do you want to be reabsorbing more sodium and thus more water via osmosis? No, right? That's going to further increase blood volume and further increase blood pressure. So AMP is going to inhibit the action of aldosterone, or essentially inhibit the release of aldosterone, right? And so all of these processes here, there's going to be less sodium reabsorption, less sodium going into the blood, less water going into the blood. This is going to result in more of that volume staying in the tubular fluid, right? So ultimately, there's going to be more urine formation, right? And so that sodium in that water that otherwise would have gone back into the blood and been reabsorbed, it's going to stay in the tubular fluid and then ultimately get excreted as urine, right? So increasing GFR, promoting urine formation. So think of these two, aldosterone and ANP, as being antagonistic to each other. They're doing opposite things, right? So this is kind of a summary slide of some of the things we talked about with relation to sodium. Again, the vast majority of it is going to be in the proximal convoluted tubule, right? Proximal convoluted tubule. Parts of it, as we'll see in a little bit, will be in this loop of Henle, right? We'll talk about the descending limb and the ascending limb and how they're slightly different in terms of what they're permeable to. Some of it in the distal convoluted tubule, and then as we mentioned with the hormones, some of it can be regulated by the principal cells in these collecting tubules and collecting ducts via aldosterone and AMP. Right. So we'll stop here for this part. We'll pick up in the next, and we'll begin talking about water reabsorption along different parts of our nephron.